Okay, so good afternoon. Welcome to the ARO seminar series session entitled Genetic Dissection of Cochlear Circuitry. As many of you know, my name is Matt Kelly. I'm a member of the intramural program at NIDCD and currently a member of ARO Council. And I'm very happy to be the moderator for today's seminar. Our presenter, Dr. Lisa Goodrich, is very excited to host this webinar for you today. And I'll just give you a little bit of introduction about Lisa now. So Lisa received her undergraduate degree in biology, summa cum laude, from Harvard University in 1991. After a year working as an electron microscopist at University College Dublin, she joined the PhD program in neuroscience at Stanford University School of Medicine. For her graduate work with Dr. Matt Scott, she cloned patched orthologs and demonstrated the importance of patch signaling in the developing nervous system and its relevance to cancer in humans. She continued to study mechanisms of neural development as a postdoctoral fellow in the lab of Dr. Mark Tessier-Levine, where she participated in a gene trap mutagenesis screen to identify novel axon guidance receptors. Since starting her own group in the Department of Neurobiology at Harvard Medical School in 2003, Dr. Goodrich has applied her knowledge of neural development and mouse genetics to the auditory system. Over the years, her laboratory has studied cellular and molecular mechanisms of circuit assembly in the spinal cord, hindbrain, retina, and inner ear. Currently, the primary focus is the formation of reciprocal connections between the cochlea and the auditory brainstem, with projects investigating the development and maturation of diverse spiral ganglion neuron subtypes, heterogeneity among oligocochlear neurons and their interactions with spiral ganglion neurons, the influence of glia on cochlear wiring and function, and the organization and function of spiral ganglion neuron inputs onto octopus cells in the cochlear nucleus complex. Dr. Goodrich was appointed professor of neurobiology in 2015 and has served as the scientific director for the Hearing Restoration Project at the Hearing Health Foundation since 2021. Before getting started today, we would like to review a few meeting tools that you as an attendee will be using throughout the presentation. These tools are located on the bottom of your screen. To keep questions from becoming distracting to our presenter, please utilize the chat function directed at myself as the moderator. And I'll be sure to get those questions asked properly at the end, well, most likely at the end of the talk. You're welcome to submit your questions at any time throughout the presentation by typing into the chat. Or you can actually, and clicking the send button. Any questions that are not addressed during the session will be answered by the presenter in a follow-up email. Closed captioning for this meeting is available for your use, and this session is being recorded and will be made available for additional viewing. We hope you will also participate in our post-webinar evaluation so we can gather feedback. Also, be sure to join us on Wednesday, November 30th at 12 p.m. Eastern Time for the continuation of this series with a talk by Dr. Rona Hersano. Finally, if you are having technical difficulties, please email headquarters@aro.org for assistance. And now, without further delay, I'm happy to turn things over to Dr. Goodrich.
Thanks, Matt. And wow, it's so great to see so many familiar faces. It's like a preview for going to Florida in February, and I hope that I'll see all of you there. So let me just get this up here. Okay. All right. Can you see the right slide?
Looks great.
Yes.
Oh, great. Okay. Thank you. Okay. So thanks very much for having me. As Matt said, like what my lab is really thinking a lot about today, these days, is not just the spiral ganglion neurons, but also kind of the other neurons that are in the cochlea that come from the central nervous system and other regions in the peripheral nervous system as well. And so, you know, I think for a long time when I thought about cochlear circuitry, you know, you stain and you look and see these beautiful radial bundles, and the spiral ganglion neurons play such an important role in all of this. And what I want to talk about today are kind of the other innervation in here, which is shown here in magenta, which comes from the efferent system. So let's see, there, okay. So let me start by just reminding you of kind of like everything that your auditory system is able to accomplish, because this puts in context why what the purpose of the circuitry is. So of course we use our ears to avoid damage, in this case, to avoid becoming food or to locate that food. As human beings, we use our sense of hearing for communicating over great distances. And I always say it's amazing how much you can communicate just with your voice, and that the minute you pick up the phone and hear your mother's voice, you know immediately how the conversation is going to go, right? And how much we have a sense of everything happening in our world and what it means to us through just our sense of hearing. And I think kind of a really beautiful example of this is our ability to appreciate music. And what has struck me for many years is the fact that we can kind of do all of this at once, right? So you can be hearing approaching danger, you can be talking to somebody in front of you, you can have music going on in the background. All of this is hitting your ear, you know, at the same time, and somehow our brain is able to separate those sounds and make sense of it. That's really an amazing feat. So where does this start, and what is the nature of the information that's initially detected? Of course, this all begins in the inner ear. This beautiful structure here. And it's, you know, this bony structure where you have to break it inside to see the goodies. And when you look inside, you'll find this sensory epithelium that's coils along the length of the cochlea, the structure down here. So the sensory epithelium, of course, is well known to all of you in this audience, is where sound is initially detected. And it's detected by these extraordinary cells here, the hair cells, which are gorgeous, sensitive machines that can respond to tiny deflections of movement that I always say kind of have only one fatal flaw, which is that they don't have an axon, right? So if your brain is going to make sense of all of this information that the hair cells are initially detecting, you need neurons to encode it. And that is accomplished by the spiral ganglion neurons, which are shown in blue here. The spiral ganglion neurons are bipolar neurons that sit right, you know, near the organ of courting. They send a peripheral process up to the hair cells, either to the inner hair cells or the outer hair cells, and then their central process into the eighth nerve and out to the brain. So how does the organization of these spiral ganglion neurons lead to coding of the information that's coming from the hair cells? Well, the kind of simplest logic that we can use to understand this is just looking at morphological subtypes. So the most basic division is of the type 1 spiral ganglion neurons, the vast majority, and those are the neurons that are innervating the inner hair cells. And really, these are the hair cells that are detecting the sounds that comprise most of what we consider to be hearing, right? And
hey,
then
Lisa.
Yep.
Lisa, if you're using a pointer, we can't see it.
Oh, you can't see it?
No.
Interesting. Let's try this. Can you see that?
Yes. Yep.
Okay. Thank you. Okay, so then the minority population, of course, is the type 2s, which innervate the outer hair cells. And the outer hair cells play an important role in gain in the system. And the type 2s may be carrying some information about pain or damage, but we won't really talk about them further today. Most of what I'm going to talk about is going to be related to the type 1 innervation, because that's really where the hearing is being encoded for processing by the brain. So that's a basic division. But then there's, of course, organization spatially that captures information about frequency, right? So the neurons, along with the hair cells, are organized along a tonotopic axis with frequencies going from low frequencies in the apex, spiraling down towards the base where the highest frequencies are encoded. So that the neurons and the hair cells up here are detecting the low frequencies, and down here, the high frequencies. In addition, there are differences among these neurons that can only be revealed really by recording from the neurons. And those are differences in their firing properties. And this was first observed in kind of these classic recordings from Nelson Kiang from anesthetized cats, where he could stick electrodes in to record from single fibers in the eighth nerve, and observed, even with a similar frequency region, all here around 11 kilohertz, different spontaneous firing patterns, where some of the fibers showed very little activity, low spontaneous firing rates, others had medium levels of activity, and others had very high levels of activity. So it's thought that kind of this variation in the firing properties, along with differences in the ability to detect different frequencies and at different intensities, together allow us both to hear the wide range of objects that we are encountered, and also to pull them out against the background noises that that they are against. So in terms of how this works, what we know is that these different types of neurons that were defined by their physiological properties actually have some anatomical properties that are very orderly and allow us to recognize them. So if you, for example, filled fibers that had lower medium spontaneous firing rates, it was discovered that those tended to project to the medullary side of the inner hair cell. That's the side that's closest to the spiral ganglion, okay? Where what my former postdoc Rika Shrestha always called the beer belly of the hair cell, and I like that, and I want it to stick. I think it's a really easy way to see it. So that's the medullary side of the inner hair cell. And these terminals tend to have smaller puncta of glutamate receptors opposite larger synaptic ribbons. Okay. On the other hand, the high spontaneous firing rate neurons project their terminals to the backside of the hair cell. This is the pillar side. And they have larger glutamate receptor puncta that are opposite smaller ribbons. So a few years ago, when Rika was a postdoc in the lab, we and other labs performed single-cell RNA sequencing that showed in a very unbiased way, and it was actually incredibly gratifying to see this repeated by multiple groups, that we all landed on the same answer, which is that there were 3 very clearly molecularly distinct subtypes of spiral ganglion neurons. There may be additional variation within these, but these were the 3 groups that everybody landed on. And that based on what we knew about the anatomical properties of the physiologically defined neurons, that we could kind of broadly assign them as the 1As were most like the high spontaneous firing rates, the 1Bs like the medium, and the 1Cs like the low. And because these were molecularly distinct, this allowed us to obtain some markers so we could actually kind of study this wiring in more detail, in that the low and medium SRs, the 1Bs and 1Cs, are positive for LYPD1 and have lower levels of calretinin, whereas the high spontaneous firing rates, which what we now call the 1As, have higher levels of calretinin. So collectively, this population of neurons is in the activity, you know, whatever they're spiking, that activity is encoding sounds in our world. And so this information is sent along the 8th nerve into the auditory brainstem for a lot of processing that all converges in the inferior colliculus and goes on up to the cortex. And so this, to me, is kind of what really keeps me motivated, which is that in the end, like, it's the activity here, right, that determines what's perceived up here. This is that's all the input. So that's nice. You know, we think about all that activity, but the fact is that while it does originate from the hair cells and there is that afferent activity, all of this actually is directly impacted by efferent feedback. And so there are efferent pathways starting all the way again up in the auditory cortex to project down into the auditory brainstem, with populations of neurons that sit in the brainstem that actually project themselves out through the 8th nerve to the cochlea, where they can directly impact the initial encoding of sound information. So if we now zoom in here and look at these efferent systems, there are 2 basic systems of olivocochlear neurons. The MOCs, those are the medial olivocochlear efferents, which sit in VNTB, and the LOCs, the lateral olivocochlear efferents, which sit in LSO, intermingled among the principal neurons there. So the MOCs predominantly project to the outer hair cells. Okay, they are mostly contralateral, but not entirely. They tend to be inhibitory. They are cholinergic, and there's a lot of evidence that they can modulate cochlear gain, for example, by inhibiting the activity of the outer hair cells. The lateral olivocochlear neurons, the LOCs, are a little bit more mysterious. They project their terminals. They end pretty much, you know, close to the ends of the SGNs. So they project onto the SGNs. They are predominantly ipsilateral. However, there's a lot of evidence that they can be both inhibitory and excitatory. And while they're predominantly cholinergic, many other neurotransmitters have been reported to be expressed by them. And in general, their function is really largely unknown, though there have been a lot of ideas put forward. So in terms of what happens out in the cochlea, as I said, this efferent feedback is intermingled. This is a really important part of that primary cochlear circuitry. The projections extend over great lengths, they branch a lot, and they are among both the SGN terminals and the hair cells. So something about this innervation is impacting the sense of hearing. And among the ideas is that this innervation could protect against acoustic trauma. Maybe it helps modulate attention, what you should, which sounds are most salient. It's been proposed that it can balance interoral differences for sound localization to help isolate speech from noise. And there's good evidence that this system can also directly impact the development of the cochlear circuits. And this is something I find very intriguing, actually, because it's coordinating development between the CNS and the PNS, which is going to be really important for having an effective overall circuit. So having spent quite a few years enjoying learning about the SGNs, we realized if we really wanted to understand both their development and how they contribute to hearing, we needed to also start thinking about the efferent system. I was lucky to convince several people in the lab to take on this challenge, because it really is a difficult project. There are not very many of these neurons at all, and it was going to require, it required a lot of persistence to figure this out. So the work I'm going to present was led by Michelle Frank when she was a graduate student in the lab. She's now a postdoc at MIT. Then she worked with a postdoc in the lab, Austin Sitka, who's still in the lab, also working on the efferents and starting to look more closely at their development. And then a very enthusiastic Northeastern co-op student, Mackenzie Hunt, who joined along and really was always this happy in lab and kept all of our spirits up when we're doing very difficult experiments. So what do we know about the olivocochlear neurons? Like I said, the first thing I can say is that they're predominantly cholinergic, which helps us because then we can use genetic tricks to look at them, right? So we can use CHAC-CRE, which labels cholinergic neurons, together with TD tomato. And what I want you to appreciate is how few neurons there are here. Okay, so there's just not a lot here. There's kind of a few hundred per brain. And the other thing I want you to appreciate is that they're right up against another population of cholinergic neurons, which are the facial branchial motor neurons. And this is both a challenge and kind of a puzzle as well, because these populations of neurons actually are very closely related developmentally. So all of these neurons develop together from motor neuron progenitors. So these neurons, these olivocochlear neurons that are impacting hearing, they're kind of, they're basically motor neurons. They're cholinergic. They're developing along with all these other motor neurons, but then they've taken on these different properties. So these motor neuron progenitors will produce precursors for all of the inner ear efferents, which will also include vestibular efferent neurons, and then these facial branchial motor neurons. And they will actually send their axons out together, kind of what will start to form the seventh nerve, right? And then the olivocochlear neuron axons, they will actually deviate and project into the eighth nerve instead of the seventh. And so developmentally, they start from a common progenitor that goes through a couple of stages of becoming kind of more brachiovisceral and then setting aside the OCNs and the FBMNs. And they share many markers that are common for other motor neurons. And the only thing we knew that would distinguish the OCNs from all the others is the expression of the GATA factors, GATA2 and GATA3. After that, there's something that is going to divide these OCNs into MOCs and LOCs, but we really haven't had any good markers or really any understanding of how this division happens or when. Once these neurons are set, however, they do extend their projections into the cochlea, and that's where they can have some very interesting developmental impact on the cochlear wiring. So already at birth, these projections have started to reach into the organ of Corti. They haven't yet formed any synapses, but the projections are there. And in data I won't show you, we can actually see cholinergic innervation kind of intermingled in the spiral ganglion during very early embryonic stages. They're reaching there quite early. A few days later, they are present during a period of synaptic refinement when the inner hair cells and the outer hair cells and the type 1 and the type 2 spiral ganglion neurons are forming their synapses. The efferents are right there. And then by about P27, we can see the mature configuration. And you can also see that during this time, there's a period when the MOCs are innervating both the OHCs and the IHCs. But we really, have not had ways to separate the MOC contribution or the LOC contribution to understand how this impacts development. So we wanted to gain genetic access so we could start to study these neurons, understand what makes them different from the facial branchial motor neurons and other motor neurons, to be able to isolate them from the facial branchial motor neurons so we could start to study their function and to start to do some clean manipulations to figure out both how they impact hearing and how they impact development of the cochlea. So after a lot of frustrating attempts, I have to say, when we started this, we thought we know how to do single cell RNA-seq. We've done this before for spiral ganglion neurons. It's the auditory brainstem is a different beast altogether, and we had to adapt all of our protocols. And I just want to kind of give a shout out to any students in the audience to say that, just hang in there. It's good to try hard things. Michelle, hung in there, and she figured it out a lot through just talking to people and being very observant. And so she became aware early on, before it became very common approach, of the use of nuclear labels to that would allow us to get around the problem of getting the cells out of kind of all of this myelin that was gumming up everything we were trying to do and just break out the nuclei. And so she used Chat-Cree to label nuclei throughout the brain. And then she and Austin microdissected out the region of the auditory brainstem that contains the whole superior olivary complex plus associated. They did not try to microdissect out. They just hoped that the genetics would take us there. Dissociated all those nuclei, then sorted out the cholinergic nuclei, which are expressing GFP with the SUN1 GFP allele, and then sequenced all of those. And so they did this from 3 different ages. They collected 45,000 nuclei from 61 animals at P1, P5, and then mature animals, so that we could follow both their development and their mature function. And you can see that we got lots of different kinds of nuclei here. And happily, among these, we were able to identify both LOCs and MOCs. So how do we find these? Well, we went back to what we knew, which is that these OCNs express motor neuron markers. So for example, if we look here on a feature map of where the motor neuron marker islet1 is, we could see islet1 come down here with all of these motor neurons. This is probably a bunch of facial in here, or some of them are going to be facial. And then out here, this population also expresses GATA3, which we knew to be expressed in the olivocochlear neurons. So again, this took a lot of persistence, because I think you can appreciate there's not that many cells left here out of our 45,000 nuclei, but it was enough to figure things out. So how did we assign which were LOCs and which were MOCs within these clusters? Well, we could go back and use anatomy, right? So I told you that the LOCs sit in the LSO up here, whereas the MOCs sit in the BNTB down here. And so when we looked at what was differentially expressed in the 2 clusters of OCNs, we saw that there was enriched expression of Col4A4 in one population. And when Michelle did the in situs, she did it on Chat-Cree labeled tissue. She could see co-localization of Col4A4 with the Chat-Cree TD tomato signal here in LSO, which is where the LOCs are, with no labeling at all in VNTB. So that told us that this really is the LOCs. And again, I just want to point out that you need this genetic marker here, because there are lots of other neurons, okay? And so this really is that it is the LOCs here, because they are the cholinergic cells there. And this point is driven home if we look at a different marker here. In this case, this is ZFP804A, which Michelle found to be enriched in the MOCs. And she could see enrichment of that signal with these arrows here in the genetically labeled MOCs in VNTB. Now, there is also signal in LSO, but it's not co-localized with Chat-Cree. So it is not specific. It's not that it's not expressed in LSO, it's that it's not expressed in the LOC. So this tells us this is a very clean markers. So what can we learn about the differences between LOCs and MOCs? So here's kind of the obligate heat map, just to say that there are a lot of genetic differences. And I don't have time to go into all of them. I'll kind of talk about a few of them that we found to be most informative. So first of all, we now have some genetic markers for MOCs and LOCs just listed here, genes that we could find that were transcription factors that might play a role in their early development or segregation. And then in addition, we found a lot of adhesion molecules that could help set up different circuitry. So for example, different coherence expressed in each of these populations. From a functional point of view, we also made a lot of interesting observations, and this has been kind of a really fruitful way of thinking about it, of how can we learn something about what these neurons do by asking kind of what kind of inputs they get and what kind of outputs they make. And so if we look, for example, at the different kinds of neurotransmitter receptors that LOCs and MOCs express, we can kind of get some sense of the kinds of responses they could mediate. So they both express AMPA receptors and kainate receptors, but only the MOCs express NMDA receptors, for example. Okay, so that would tell us that, well, they both respond to glutamate. The MOCs are responding differently to glutamate than the LOCs do. They are cholinergic, but they also express acetylcholine receptors, but they have different subunits. So again, they're going to respond to acetylcholine differently. Okay, and then finally, and these are some of the differences I have found kind of most intriguing, is that they can express different receptors for some peptides and hormones. Sorry, I forgot about the inhibitory stuff. So different GABA and glycine receptors too. So for example, only the MOCs express the opioid receptor, whereas the LOCs actually express the orexin receptor. And so I don't have any answers yet, as to what any of this means, but the nice thing about these kinds of single cell RNA sequencing databases is it's a really good place for some hypothesis generation. And I'll say that all of this data is now available in a manuscript on bioRxiv, and I hope that those of you who want to dig through it can find things that will stimulate your own experiments. So that is kind of like some basic difference between the LOCs and MOCs. And we have markers to distinguish them from the facial. But what we've been, as I told you, the thing that we've been kind of most puzzled by are the LOCs and what the heck they're doing. And one of the things that's been challenging and thinking about the LOCs is that they themselves appear to have some additional heterogeneity. So for example, there are some that express dopamine or produce dopamine. There's some outside the LSO, some inside the LSO that express dopamine. And then in addition, they have wide-ranging morphologies, right? So sometimes they'll have very long projections, sometimes these kind of nests of projections. And then in addition, while they are largely cholinergic, as I said, there's been some of these, they can express dopamine, and this can actually be dynamic, so that Elizabeth Blavatsky's lab showed that actually sound exposure can cause these neurons to change their neurotransmitter profile. So there's that kind of heterogeneity, and that there's a number of other transmitters that have been reported and shown to be expressed in subsets of LOCs. So we were curious whether we would be able to find any molecular differences that might help us understand both what the LOCs do and kind of the basis of this heterogeneity. So to do this, Michelle subsetted just the mature LOCs and MOCs. So this is from 32 animals and then 589 nuclei. And she got 5 clusters. So we can identify these as MOCs because they express that marker I told you about. That's an MOC marker, and it's very clean. These are clearly MOCs. We have tried really hard to understand what the second cluster of MOC2s might be. It's got so few cells that we don't know what it is. And if you guys have any thoughts, happy to hear them. We have pretty much ruled out every technical explanation we can think of, but just to say we haven't been able to figure it out. This population over here, the LOCs, okay, here, and you can see this actually came into 2 very clear clusters. Again, there's a miscellaneous cluster that's too small for us to figure out. So let's just look here then at these 2 clusters that really came out robustly and think about what does this tell us about differences among the LOCs. So if we look at what drives differences between this cluster here and this cluster here, this is not in the kinds of gene expression changes that we see are consistent with the idea that these are actually different cell types. So why do I say that? Well, first of all, we see differential expression of some adhesion molecules, such as the tennurons and FA6. So for example, FA6 is expressed only in this one cluster, which we're calling LOC1s, and it's not in this other cluster, LOC2s, okay? Likewise, we see differential expression of tennurin 3. This is very common and actually has been reported by Leach and Lowe as a very good indicator of cell type, right? That you have different transcription factors and adhesion molecules. So this makes us think that these are probably really different types of LOCs. So what would you imagine different types of LOCs could do? To figure this out, Michelle compared what's different between the LOC1s and LOC2s. And it's shown here in a volcano plot where genes that are more highly expressed in LOC2s are on the right-hand side, and those that are more highly expressed in LOC1s are on the left-hand side. And what jumped out to Michelle was that the genes that were enriched in the LOC2s tended to be neuropeptides. And so this is just shown again as violin plots here. So for example, the LOC2s uniquely express urocortin and neuropeptide Y, and they have the highest levels of CGRP 1 and 2, so CalC and CalSP. So we wanted to figure out, well, where are these like anatomically, or like what does it mean to be LOC1 and LOC2? And this gave us further evidence kind of supporting the idea that these might really be different cell types, because when Mackenzie Hunt, when she was in the lab, looked at the expression of these peptides, she found that they were enriched in the medial wing of the LSO. So you can see a gradient of CGRP where it's highest medially. We have an MPY GFP line where you could see really those MPY cells are in the medial wing, staying for urocortin, and it's all there. And this is just a quantification of the overlap of these kind of various peptidergic neurons that really the best marker for them is MPY. Almost all of them express MPY. So we wanted to see kind of, well, where do these project in the cochlea, and what do we know, what can we learn about them from their anatomy? And so the nice thing is that there are a lot of tools for MPY, and there's also a lot of tools for chat. So we took advantage of these tools to do an intersection where we use chat Cree together with the MPY flip line to turn on a reporter that requires both Cree and flip. Okay, and so this would mark just these neurons right here. Okay, and we can see that those are labeled only in the medial wing. Okay, so the MPY positive cholinergic neurons are in this medial wing. Further, if we look at the onset of MPY expression, we can see that as early as P0, you already see this population limited to that medial wing. And the fact that even in adults, they're still limited there suggests to us that this is a population that's set aside pretty early in development and is anatomically restricted. So that's sets us up to ask, well, how might these projections be interacting with the circuitry out in the periphery? So for one thing, if they're in the medial wing, we know that there's a tonotopic axis. Is this a population that only has anything to do with, say, high frequency neurons? Or is there some other logic for how it interacts with the spiral ganglion neuron circuitry? Because remember, the LOCs do interact with the terminals of the SGNs. So to remind you, we have 3 different types of spiral ganglion neuron projections that are segregated along the modular pillar axis. And so we can now use, for example, differential expression of calretinin to look at the different subtypes of SGNs and ask, how do these LOC2 axons interact with that, those different subtypes underneath the inner hair cells? So we can use this genetic labeling and first look to see where are these projections in the cochlea. And the first thing that Austin Sitko, who did these experiments, observed is that despite the fact that the cell bodies are restricted to the high frequency regions, their axons project along the entire length. So she could see terminals. Yes, there were more of them in the base, but they reached all the way up to the apex, to the low frequency regions. Further, if we now look at how they're interacting along the pillar modular axis. Okay, so here we're looking in 64 kilohertz region where they're in green, and in magenta we've labeled the hair cells and spiral ganglion neurons. We can see that there's there's maybe more of them on the modialar side, but they're also present on the pillar side. And if we turn it on the side to look at that, you can see that they're really present on both sides. Okay. So this suggested to us that whatever this population is doing, it has wide-ranging effects because the axons are spanning across many frequencies and they're interacting with both the pillar and the modialar side. But it didn't give us a sense for whether kind of individual axons might have some stereotyped circuitry or morphology. So for this, we really needed to get into the single cell level. So here we were able to take advantage of an observation that Michelle made early on, which is that both MOCs and LOCs express the receptor RET. And there was a RET-CREAR tool available in the colleague of our lab down the hall, David Ginty, which he had shown to work very nicely. And so if we inject tamoxifen to sparsely induce activity of RET-CREAR during development, Austin found conditions where she could get very nice sparse labeling of individual MOCs or LOCs, in this case, just recognized by their morphology and by their terminals. So Austin set out to do a lot of reconstructions and try to work out, was there any logic to how each of these populations interact with the rest of the circuitry in the cochlea. And so let me just walk you through what she did first for MOCs, because I think it's kind of maybe more intuitive to look at because we know a bit more about the MOCs. So the MOCs form these very stereotypical terminals amongst the outer hair cells. And this has been known for a long time. And so Austin could get these GFP labeled MOCs, okay? And then she would stain for synaptophysin to identify presynaptic puncta, and then calretinin to identify both the hair cells and different types of SGNs. She can use the mask of this GFP labeled process to identify just those puncta that are inside the terminals, okay? And then create a reconstruction where we can identify the puncta, the presynaptic puncta, and take a guess as to their postsynaptic target. You run out of channels, so there's only so much you can do here. So first of all, we saw morphologies that were expected for MOCs with puncta where we would expect to see them amongst the outer hair cells. But in addition, we also saw puncta down here in the interspiral bundle where actually SGNs are interacting with inner hair cells. And this was actually really gratifying because this fit with some recent EM studies reporting that MOCs, in fact, do make synapses with spiral ganglion neuron terminals. And so this gave us a lot of confidence that our method is very very sensitive and accurate. And let me just show you kind of a movie. Oh, now I have to change my. How am I going to do this? I don't know how to get it back there. Okay. Is the movie not playing? All right. Well, you're not going to get to see the movie. I don't know why it won't play. It played on my rehearsal. Let me just go back. No. Oh, well, okay, that's too bad. Anyway, the reason I wanted to show the movie is that this looks very flat here, but we can actually look at it in 3 dimensions and also get a sense of position and be very confident about where those puncta are. And in fact, I just wanted to say that this presence of presynaptic puncta that are opposite spiral ganglion neuron axons or processes, we saw that in 18 out of 40 of the reconstructed MOC. So this is not rare. All right, so let's move on to the LOCs and let's see if this movie will run. All right, too bad. Okay, so I'll just show you the breakdown of it. But in this case, now we're labeling an LOC. It's been stained for synaptophysin to label the presynaptic puncta. And in this case, we've also stained for MPY so that we can capture that peptidergic population. And then for calretinin, again, so we can see the hair cells, but also to be able to distinguish the different types of spiral ganglion neuron terminals underneath the hair cells. And so what Austin can do is, again, reconstruct individual LOCs, use that as a mask to separate out those synaptophyes and positive puncta that are in this LOC, and then reconstruct that and assign for each of those synaptophyes and puncta whether that is touching a CALB2 positive SGN process or is touching something that does not express calretinin, which we would infer is probably a 1C. Okay, all of them are amongst the spiral ganglion neuron terminals. And when you study for MPY within these MAS1s, the first thing that jumped out is that it's actually itself very heterogeneous. While this is a peptidergic LOC, the MPY is not present at every synaptophyes and puncta. Okay, so some of them will express both synaptophyes and MPY, but others are MPY negative. So Austin went through very systematically and reconstructed, in this case, I'm just showing this reconstruction here, many LOCs, and then assessed what is the nature of their presynaptic puncta. Are they contacting calretinin? Do they express MPY? Are they on the pillar or the modular side? How long is the projection? How many frequencies does it span? And she did this for many, many LOCs and saw an incredible range of morphology, right? So some of them turn to the right, or, turn towards apex, some turn towards the base. Some of them have these like big nests. Some of them have these kind of short little projections. Some of them bifurcate and go in both directions. Some of them can be 100% opposite calretinin positive fibers. Some of them can be 100% MPY positive or only 50% MPY positive. We basically saw every combination, and Austin has looked at every possible kind of property that we thought could possibly, vary in a way that was informative, and found no correlations that were strong or predictive, in that the MPY positive fibers could turn to the apex, the base, or both directions. They spanned a wide range of frequencies. You could have, whether they had MPY, this is an MPY negative, and this is MPY 100%. Either way, they could have few or more synaptophyes and puncta. There was no evidence that the MPY positive puncta had any different selectivity for whether they were opposite a calretinin positive target or not.
Hey, Lisa, I think your pointer has disappeared again.
Oh, because of my laser thing. There, okay. It's okay. All I was pointing out is that there's no correlation that's strong or predictive. So this is actually probably, like, while we love to think that like any molecularly defined subtype is going to have some kind of beautiful anatomical prediction, it's really more common in the nervous system. It's just harder to make sense of, right? That these neurons, or the individual axons, just don't seem to be highly selective, but it's just part of what they're doing. So we wondered, well, maybe the differences are actually more physiological. And for this, we teamed up with Kat Weitz at the NIDCD, and we sent them some CGRP GFP mice, where we could use that gradient of CGRP to identify kind of the high peptidergic neurons in the medial wing versus the lower ones kind of nearby. We decided not to go all the way across the tonotopic axis. We wanted to kind of stick to one tonotopic region so we could disentangle the effects of peptide identity from tonotopy. And so Kat's lab performed both voltage clamp and current clamp, and kind of came to the same conclusion that Austin did, which is, while there's a lot of variability, that variability does not correlate in any predictive way with MPY or peptide status, right? So in this case, looking at the levels of CGRP. So they measured, how bright basically the GFP signal was in the live tissue, and found, like I said, a wide range of properties, but zero correlations. So this leaves us a little stuck, and makes us think, it's kind of like hard to think, well, what could these different populations be doing? And so we thought, well, when do these differences even come about, right? So when is this peptide status changing? And so when Michelle went back and looked at that developmental single nucleus RNA sequencing, she saw that kind of across the board for these peptides, they were coming on quite late. That some of them were present early, but that these differences were coming up later in development, so by P28. Further, when Michelle did in situs, for example, for urocortin, she could see that early on, urocortin was expressed, but it didn't become restricted to the medial wing until after the onset of hearing. And this was also true for CalC and CalSB, where she could see kind of these broad expressions patterns that got higher as development proceeded and then were restricted to the medial wing only by P28. So the fact that this restriction happened after the onset of hearing made us wonder if it might actually depend on the sense of hearing itself. So Michelle got animals from our neighbors next door, the David Corey's lab, where they had TMC1 and TMC2 mutant mice breeding. And she looked at the expression of her peptide, of the various peptides in the LSO. So first, let's look before the onset of hearing, so P7, P8. And she could see, we're just showing the neurons here by chat. This is where the LFCs are. You could see that you've not yet got a gradient to CGRP. It's not any different in the mutants. And that you see this onset of MPY that's still medial. It's not any different. Okay, and she quantified this extensively. I won't show you all the statistics, but everything is flat. We saw no differences prior to the onset of hearing. However, it's a different story when she looked in the more mature animals. So now we're looking around P27, P28. What she found is that the CGRP gradient no longer matured. So it basically doesn't become restricted, instead stays high across. And the same was true for urocortin. And this is quantified here, where she looks at there's no change in the fraction of CGRP, but there is a change in the slope because the intensity is higher out here laterally. And there's a significant increase in the fraction of urocortin positive cells out here. We can see it stretching out much more laterally. So the fact that these peptides were sensitive to the hearing experience made us wonder if they could, like dopamine, also be induced in mature animals by intense sounds. And so in this case, we took animals that were either unexposed, just put into the acoustic trauma box and handled in parallel, or else exposed to 2 hours of 8 to 16 kilohertz noise for at 97.5 dB for 2 hours, I said. And then we looked at dopamine just to reproduce the previously published results, urocortin, CGRP, and MPY. And what we saw was, yes, we could see induction of the dopaminergic population. We saw this massive expansion of urocortin, this increase in CGRP, and a bit of an expansion of MPY, where it's still medially restricted, but it seems to expand a little bit. And I want to just kind of stay here for a little bit and make the point that this population that's medially, that there can be both an MPY population that is defined genetically in the medial wing, and that MPY can also be part of a cell state, but that this is still consistent with our interpretation because we never see it induced far out here in this very far part here. Okay, so here's all the quantification where we see this induction of TH positive LFCs, many more urocortin positive. We don't see a change in the fraction of CGRP positive because it's just the slope. And then we see this increase here in MPY. And so collectively, this has made us even more curious about what all these neurons are doing. And we have multiple people in the lab working on this right now. And we will actually have some data on this to share at ARO if you're interested in looking at it. We have all the genetic tools and we're trying to get in there and do some manipulations. But it's also kind of raised some very interesting developmental questions, because as you can see, these fibers are just kind of like intermingling with everything in the circuitry out there. And it's very curious to think about how, is this purely opportunistic or are there actually developmental rules, and how do all these different adhesion molecules in fact help sort out the circuitry? And these are things that are for the future. So I will end by acknowledging my wonderful lab. It's just a pleasure to work with all of them every day. And I don't know how many of them are listening. I told them not to today, but I think they might be doing that anyway. In any case, I was particularly grateful to Michelle for taking this on and being persistent when it was extremely difficult, and then for being so enthusiastic about it. She managed to get a lot of other people to join the project, and it's a big part of the lab now. Mackenzie for the work she did. I failed to mention, I'm sorry, that Caroline Yuck did all of the MOC reconstructions, and that was a really important contribution to the project. Thanks to the Northeastern Co-op program for continuing to send us wonderful undergraduates to work with us. Really grateful to Kat for this collaboration. It's so challenging when you get no correlations, but it was interesting that was just, what we saw both anatomically and physiologically. To Bernardo Sabatini for making his 10x Chromium platform available to us. And then, of course, the NIDCD for their support and funding, and to additional sources of support for our work. So I'm happy to stop there and take some questions.
Okay, so Lisa, thank you. That was fantastic. Really nice work. There's one question in the chat so far, and that is, so the first question here, does this sound activation have a relationship with the presence, I assume that's the one you just talked about, with the presence or absence of tinnitus or hyperacusis?
Yeah, interesting. I think it's an interesting idea. I don't know. Yeah, I mean, it's things to keep in mind as we move forward with this work.
Okay. Next, do you have any thoughts about how chronic hearing aid use or cochlear implant use might affect development or distributions of OCN?
Yeah, well, people may remember that we did find previously that we got changes in the mature, profiles of SGNs in the TMC1, TMC2 mutant mice. And I've always wondered how that might impact kind of the cochlear implants, whether the ephrains will have a similar effect or not. We don't even know what they do normally, so it's hard to kind of make any predictions. But I guess it's something, activity dependent development is a real thing. And I think it's something really important to consider in anything to do with cochlear implants.
Okay. Yeah, so that's great. And I'll ask one while we wait for some more. And that is, so it seems like your system's pretty well suited to maybe use an intersectional deletion approach, right? To maybe express DTA or something like that in these neurons that you care about. But then how will you go about trying to figure out what they do once they're gone?
Yeah, well, so we have had many coffee conversations with people about this. So we're starting with the obvious, which is most people would predict a role in protection from acoustic trauma. And so that's the first thing that we're doing, that we have done, and we've reported these results at like the Gordon conference. We've presented these results that we do see if we selectively ablate most of the LOCs, about 2/3 to 3/4 of them, that you can get more damage to the cochlea with sound exposure. Okay, now the bigger question, well, okay, but what are they doing? There's a postdoc on the lab, Gabe Romero, who is very interested in this question, and he is going about it, systematically. We don't have answers yet, but we're basically trying to use what we can think about from the genes they express and then the connections they make to come up with some predictions. There's been an idea out there for a while. I know, like Amanda Lauer's put this forward a lot, that this system could be recruited by stress, and that there's something about how to protect the cochlea from stress, either with kind of feedback, and so we're trying to come up with ways to explore that. And we've had fun talking to Amanda about this as well.
Okay. And a quick follow-up, and I'll get to some more questions. In terms of attentional responses, would you expect that the mice would have more difficulty hearing sound and, tracking sound and noise in background noise?
Yeah, I'd love to do those experiments. We've tried to do them like in other manipulations, and it's hard to get they're hard experiments. Yeah, so I think if we had exactly the right paradigm, it would be really interesting to try to do that. Trying to make sure we do it the right way.
Okay.
Yeah, and you could also think about like binaural things, right? And so we've got some things that we're considering. I'm not trying to be cagey here. I just don't want to commit to anything because we're trying a bunch of things and I don't know what's gonna come out of it. I think they're really, I don't know, it's a really interesting population. And the fact that they're producing all these neuropeptides really makes me think that a lot of it is, going to be slow acting over very, like these projections are so long. It's clearly not like, it's not something to do with like heightening frequencies, right? It's something that's overall long, slow acting effects. The other thing I forgot to mention is that this induction of the neuropeptides, this is like a week after sound exposure. This is a long lasting effect. So that makes it, it makes it even harder to think about like what is changing in the state of an animal that's hearing kind of over those time frames. But I don't know.
Okay.
Isn't that why we go to lab every day, is to do hard things?
Yes, absolutely. Okay, we have several more questions in the chat. The first is, you showed NMDA receptors. I was only familiar with ACH and glutamate. Does that mean some learning is implicated in the auditory system neurons?
I mean, it means that there's some plasticity, right? So, I don't know that it's learning per se so much as, that the system is likely adapting to signals that are coming in. And I think that makes a lot of sense because, there's a lot of central circuitry. And I was trying to make that point at the beginning, is that this is not all just a, one simple, just in and out reflex, that there's probably an opportunity for a lot of input from other regions of the CNS over different timeframes and different situations. So, maybe the NMDA is going to affect, responses kind of in response to other things happening in the state of the animal. I don't know.
Okay, thank you. All right, and I realize I can be reading the names on these. So Katie, this is from Katie Kint. Do you imagine there's a similar or related innervation to the vestibular system?
Yeah. Yes. The vestibular efferents. It's like we spend a lot of time staring at our data thinking, do you think they're there? Like, would they have been in our dissection? They're located a little bit more dorsally. I hunt people down who know about the vestibular efferents and say, look at our data. Tell me which ones do you think they are. We keep thinking maybe we'll figure it out. Maybe maybe that miscellaneous population, maybe that was vestibular efferents or something. It wasn't, but we just haven't been able to come up with enough markers that would kind of take us there. But we're always thinking about it, and if we figure out where they are, I'll be very happy. But I mean, the other point I would make is that this kind of efferent feedback is present in just about all these hair cell mediated systems, right? It's not just a cochlear thing. It's pervasive.
Okay. All right, next one is from Doug Epstein. Does genetic variation in LOC specific genes associate with auditory phenotypes in humans?
Haven't looked at that. It's an interesting thought. I like that. Yeah, haven't looked at that yet. We can add that to the list. And yeah.
Okay. From George Ordaway, while there were no correlations with the efferent connections back to the cochlea, do you think there might be correlations with the afferent input these neurons receive?
You mean the inputs onto the LOCs?
George, do you want to decloak?
Yes. Okay. Yeah. Underway, trying to figure it out.
Okay. From Samantha Davis, have you seen any evidence of the NMD expressing OCNs inducing synaptic plasticity, either in the form of LTP or LTD?
Haven't tried. There are probably people here who may know better than me and have tried it. We do. I'm very excited about this. We do have an ePhys rig in the lab now, and I'm starting to understand how to look at that data, but we have not ourselves done that yet.
Okay. From Kathy Sung, thank you for the wonderful talk. You mentioned that in noise-exposed animals, CGRP and NPY expression were more spread out in the OCN compared to unexposed animals. Do you know if the expression pattern would look different if you exposed animals to noise of restricted frequencies? In other words, would you expect to see different expression patterns if animals were exposed to 8 kilohertz versus 32?
It's a great experiment. We haven't done it yet.
Okay. Elizabeth Siebel, do you know what makes cells competent to upregulate NPY in response to noise damage? Do you know anything more about the properties of these cells that can dynamically change their neuropeptide expression based on state?
Yeah, we do not. And hi, Elizabeth. Elizabeth is a former student in our department, so it's nice to hear from her. So we don't know, but we are actually looking right now to see, is that upregulation? Remember, when we looked at that upregulation, it was a protein stain. And I think we don't know yet whether the RNA is going up or whether this is kind of unveiling some, maybe it's a local translation thing or something like that. So we're kind of considering that now. Any of this kind of neurotransmitter switching is poorly understood right now. And so it's something we're thinking about. And right now we're just down to, is it happening at the level of RNA or protein?
So do you think this is sort of a bizarre corollary? One of the things we found from our single cell studies was that NPY is really strongly expressed in inner pillar cells. Do you think there's any connection there between?
And you know what we found in our glia sequencing is it's also really strongly expressed in the cochlear glia. There's NPY everywhere. And, and I think I haven't figured out, we've looked a lot at where the NPY receptors are, and it's not like they jump out on the SGNs. We've tried really, we've looked at CGRP receptors, we've looked at all of the receptors trying to figure out what's the logic, and nothing has been obvious. And we've even wondered, well, you've got so much NPY around, could some of it be being taken up, endocytosed or something like that? And so maybe that adds more to the heterogeneity. I think it's really curious, and will keep us busy for a while.
Okay.
Yeah, but I also, I mean, I've seen that, because if I go into the gear and search for NPY, it's the pillar cells show up, and it's why?
Yeah, we get great labeling with that same NPY Cree that you guys have.
Yeah.
Okay, that is the end of, oh wait, one more from Yuvraj Yoshi. Thank you for the great talk, nice findings. Do you have any thoughts on innervation pattern of LOCs on inner hair cells and spiral ganglion neuron subtypes along the tonotopic axis?
Well, it was any thoughts about innervating the different subtypes? I mean, we don't see any correlation in that, an individual fiber is clearly innervating multiple subtypes. So we find, there are more lateral efferents on the modular side. But an individual fiber, if my movies had played, can have branches that go on both sides, right? And what's cool also is that it really perfectly correlates with what calretinin, right? So the calretinin thing is real. They are really restricted. The calretinin high are really restricted to the pillar side. It's quite striking, but we just don't see any evidence for any kind of selectivity. Whatever this system is doing is it's not, its effect is just much more diffuse, I would say, than the way we're used to thinking about, hair cell to neuron does this.
I wonder if there are ways to change the experience, that initial experience period, the critical period for defining things, and try and change the pattern based on the type of noise the animals are raised in or something like that.
Yeah, that's